Right, so we are going to start with the last session with Edouard Hannezo from IST Vienna, and he'll talk about bulk actin dynamics, which drives active phase segregation in the Zebrafish oocyte.
Thank you very much for the invitation. It's been a really fun day. So indeed, I'll talk about some work I've been doing in my lab, which is mainly the result of a collaboration with C.P. Eisenberg's group, and I'll, of course, acknowledge everyone in the middle. But I'll start, as sort of Thomas suggested, by a slightly more general introduction about what we mean by self-organized morphogenesis and what actin dynamics has to do with it. So, this was already covered in the past, in the previous sessions. We're all interested in morphogenesis, but one of the problems of why it's technically challenging and theoretically challenging is that it's deeply multiscale, and there's at least tens of magnitude from where the information is encoded, molecules to cells to organs, to where is the output. And that bridges over 10 orders of magnitude in total. And of course, despite of this sort of dazzling complexity and kind of length scales, you still want to ask, from the point of view of theory, whether you can extract some general design principles on how these scales communicate between each other. Of course, so it's a pretty tough problem a priori. There could be so many components, so many scales to consider. But I think one of the things that's been fascinating in the past decades from developmental biology is that actually, even though you might think that there's millions of blocks you could do things from, the picture that has emerged is a bit more that there's a rather limited number of blocks that seem to be reused in very different ways to achieve different types of morphogenesis. And therefore, one of the fascinating problems is that you have this type of blocks, and they have two properties, a few type of blocks, and they have two properties. One, they need to be able to do something very precise in a controlled way, but they have to be versatile enough that another organism is going to do something completely different via a different sequence of event. And that's really how you go from this to either of those is a super interesting theoretical question that many people, of course, have been asking. And in a way, this is probably one of the oldest questions of developmental biology, even going back to the most classical embryologists from the end of the 19th century, when they played with ablating cell lineages and seeing what happens. One of the most classical results is that you can find either extremely deterministic and intrinsic blueprints or self-organized models of development, so that you can really have two extreme ways to do morphogenesis. There are some where if you separate, like in tunicates, we can separate different types, different cells in the early embryo, and these basically do the tissue type they're supposed to do irrespective of their neighbors, so it seems like they're unfolding deterministically a sequence of events without caring so much about the neighbors. Whereas if you do a very similar experiment in other organisms, each cell is somehow going to know that it doesn't have the proper neighbors anymore, is going to react to this, and is going to do morphogenesis in a much more plastic, adaptable, self-organized. And of course, this is probably what has attracted the most attention from theorists as an example of self-organized development. So in this kind of picture, you can go back to the Lego idea, right, of these building blocks you can play with. In the example I've shown you was clearly in this deterministic way, right? You have a roadmap that arrives with your Legos, and you have to assemble them, and you deterministically assemble your Legos in a way that makes us the final structure. But you could do something else, right? And that's something that has been done actually in the past few years. You can take Legos and you can just throw them in a washing machine and see what happens, right? And after all, you could imagine sort of much more self-organized, sort of a self-assembly type, where this is going to give rise to some structure, right? So people did this proper experiment where you take Legos. There's this rather distinguished professor that sort of looks at, watches over the experiments, and then you can look at what happens in the end, and it does give rise, of course, to some structures, right? There is some spontaneous structures that emerge from this, but they're not very robust and they're not very complex, right? And the point is that, you can replace maybe a washing machine by a young infant, right? And you might think that this is a statistically unlikely event that will come up out of this washing machine. And of course, the difference is that cells are not Legos, and energy is not just inputted at the level of the washing machine. There's actually much more precise things that takes place inside the Legos to allow for these statistically unlikely event to become the actual realization. And so, broadly speaking, so my lab is really interested in this at sort of all scales, and in particular at bridging scales. And we've been super interested in looking at the questions of how much stochasticity you get at different stages of development. So trying to connect both the cytoskeleton to cellular scales, but also looking at the stochasticity at the level of single cell fate choices, as well as how you go from cells to large-scale organs, for instance, in branching morphogenesis, and what kind of simple rules can allow for self-organizing properties across scales. But today, I would like to show you a story that we've been working on in the past year and a half with the lab of C.P. Heisenberg, which was recently sent, and which has to do with exactly this question of how much self-organization and how much control do you need to put in morphogenetic event in morphogenesis to give rise to the robust to something robust. In particular, this question is going to be around the zebrafish oocyte. And how the cytoskeleton and the zebrafish oocyte helps to organize the earlier stages of morphogenesis. So when I talk about cytoskeleton, actually I'm going to talk only about actin, and I'm going to cut on this introduction because Eva already said everything that you need to know. It's one of the main force-generating structure in tissues. Of course, it's extremely complicated. We don't want to model all of this complexity. And the advantage is that in the past decade, there's been, before going to the theory, one of the interesting things, right, is you start with a structure which is microscopic, and it has been shown by many groups, including many in the room, that all of these actin filaments can self-organize into extremely complex and extremely diverse structures which power many morphogenetic events. So you can imagine steady-state spatial patterns. You can get temporal oscillations. You can get a complex mixture between temporal and spatial oscillations. And that seems to be a pretty general and extremely versatile way that you can make a huge amount of different outputs from pretty much the same components. And this is really a big question, right, of how you go from the microscale to the mesoscale to give rise to this complexity. And so theoretically, a lot of the framework has to do with simplifying the problem and dropping a lot of variables which you could put to model this kind of actomizing behavior. And so the general idea behind this, which was developed in particular by Jean-François, is to say that, of course, there's a huge amount of variables in this kind of actomizing, but actually most of the variables you don't really care about if you're imaging. In normal imaging, let's say, anyways, you're only looking at smooth average over some micron scale. So actually, what you're mostly interested about is a very small number of variables, such as actomizing density and velocity. And the advantage of just concentrating on a few coarse-grained variables is that suddenly there's not so many parameters to consider. You reduce your parameter space to billions to a few observable ones. You can think, for instance, that you might need to consider turnover, conjectivity, friction, viscosity, but and you can add a few, right? But that's a countable number and sort of manageable number of parameters from a theoretical perspective. And you can make very simple models of this, and so this has been looked quite intensively in the past few years. And you can write simple equations putting these minimal ingredients into some model of actomizing networks. And the interesting thing is, as soon as you write the most minimal model, you have a very simple route towards patterning that emerges, and this route is just contractually driven self-reinforced flow. And the general idea is that as soon as you have a bit more actomizing here, because actomizing is under tension, high density, high tensile regions are going to bring towards them, are going to attract low density region. And if you have this feedback loop that goes where things from a low density flow towards things at a high density, that's destabilizing, right? That's an anti-diffusion term, and therefore you have this self-reinforcing loop between ectomizing tension and flows, which spontaneously breaks symmetry, although it's resisted, of course, by a few things. And that's has been looked at by many groups, including, again, in many different settings, including many people in the room. But you can ask, okay, that's nice and that's nice. No, but so what are these flows good for? One thing you can do is you can do patterning, but you can also extract forces from them. That has been looked at in the context of cell migration, for instance. But I would like to show you sort of a simple way that we thought it could be harnessed in in a slightly different way as before, which is to do phase segregation. So why am I talking about phase segregation? Let me just do a tiny bit about this, and then I'll connect it to why we're thinking about actomizing in this context. What we got really interested in is these earlier stages of zebrafish morphogenesis. And if you take the oocyte, initially the zebrafish oocyte is a mix of fluid and yolk granules, so which end up as an energy source. And from between the 1-cell and the 8-cell stage, what you have is essentially a phase segregation event where all of the fluid streams upwards, and you end up with basically two phases, one phase which is pure fluid at the top, and one phase which is pure granules at the bottom. And so we really wondered, okay, what are the forces involved, what are the forces or the chemistry involved in this kind of phase segregation? And this is really, this is really the whole PhD project of Cheyenne, who's a PhD student in the lab of CP, and it's a particular collaboration with a postdoc in my, Shirley Shui, a postdoc in my lab, who did the theory on this. So let me show you first a movie before going back to Actin on how this happens. So this is so what you're going to see here is some marker for the ooplasm, which is initially pretty mixed in between. And what you're going to see is these phases of streaming, which are you get oscillations and streaming. At every time you get a cell cleavage, you start having a phase of fluid. That's the second phase. Now it's going to rest for a while. Nothing happens. There's a third phase. So that's connected to the cell cycle. And after around 4 phases, you end up with something which was mixed to something which is fully segregated, right? And why do we care? Well, I mean, we don't only care about zebrafish. This type of cytoplasmic streaming and reorganizations are key features in many events of early embryogenesis and are important for the patterning of this whole thing. And again, because this is an old question, there's been quite a bit of experiments and theories done around it. And one of the key model on this was to observe that in the phase of streaming, you get a lowering of cortical tension here, which almost looks like a bleb, and therefore this deforms at the animal pole. And the idea was that, well, if you have a deformation, you get suction of some pressure of some fluid, so maybe the fluid starts going towards there, and then you reinforce it so it stops. But then you're going to do different cycles of decreased cortical tension, which are going to suck in periodically via this geometry effect from the cortex the entire fluid. And to begin with, Cheyenne did a pretty elegant experiment to test this, which is to put the embryo in a box, in a square box, and confine it so that you can even break the cortex. So now, essentially, in this kind of embryos, which is minimal assay, it's mainly bulk. So you don't have any change of geometry, and the cortex is essentially destroyed. But if you run the movie, you still see this oscillation that happens, and after each phase, that's the first phase. The second one is going to suck in a bit more, and essentially, if you compare the global dynamics of phase segregation, it's tiny bit impacted, but basically the entire phase segregation happens as a bulk property, where you cannot really argue for anything linked to cortex or geometry. One of the interesting things that you might see is that before what does happen before each phase is that you start having actin going up at the top, right? So here actin goes up, and that's exactly around the time that cytoplasmic streaming starts going up as well. And so you might think that the bulk actin in the cytoskeleton might have something to do with this process, given the strong correlations that you get with this. So what we have so far is we're looking for more bulk explanation where there's some correlations between actin intensity and flows. And indeed, if you image it with much higher time and spatial resolution, when you get streaming happening, you start having an assembly of a very dense actin mesh, which starts flowing upwards. And so the actin starts flowing upwards, while the granules in net start flowing downwards. So you get this very simple kind of picture where at each cycle, you get actin and water going up and granules going down through this kind of bulk effect. So from there on, we asked, okay, I mean, there should be some, what kind of theories can we write? How can we understand this? And essentially, from a theoretical perspective, we wanted to answer 3 questions. One is that, okay, let's imagine I have actin flows. Let's take these actin flows as inputs. How do they translate into a plasma flow, right? Because why is it not what breaks symmetry between the fluid and granules in here? The second thing is, okay, if we manage to answer this question, of course, we just displaced the problem. We have to understand how actin flows are generated themselves. That's the second question. But also, even if we know how actin flows are generated, we need to know what says the directionality, right? How do they know to always go upwards during this process? And I'll just go through a little bit the key steps of what we've been doing. What we did from this is a very minimal model, actually, where you really need to consider 3 phases. You need to consider the actin, the actin terminizing as an active gel. You need to consider fluid, and you need to consider granules. And what we did is to write systematically in an active gel theory a 3-phase model where you have actomyosin as an active gel, which is embedded in a passive medium, which is a poroviscous medium of fluid and actin, and we wrote rigorously all of the couplings, all of the frictional couplings between all the 3 phases that you can get, as well as the different rheologies of all 3 phases. And you can write this, of course. But the simple intuition you can, you can solve this kind of model analytically. But the intuition you can get, which is pretty simple, is that so you're going to have frictional interaction between all 3 phases, which are just passive frictional flows. And the general idea is that you have a friction between actin and fluid and water, and a friction between actin and granules. And essentially, the one which has the highest friction is going to flow with actin, right? Because not everyone can flow up because of incompressibility constraints. So the general idea is just you need to know the relative frictions between all phases to know who flows with who. And indeed, so you can come up with a simple analytical expression for this, where you compare the velocity of actin and your granules versus the relative friction between the two, and you can either have actin. If actin goes up, you can either have fluid going up as well, or granules going up, which would give rise to the opposite polarity. Okay, so what is this friction between phases? The advantage is that for simple passive friction, we can calculate. I mean, in terms of scaling, at least we know where it is. So if you have something like a polymer inside a fluid, the friction between the two is effectively proportional to the viscosity of the embedded fluid divided by the mesh size of the polymers, right? So if we have the viscosity and the mesh size between each phases, we have at least a scaling order, an estimate of the different friction coefficients. So that's what Chaire did. Actually, I'm not going to detail all of this, but he tried to estimate as many parameters as he could. So he measured conductility through laser ablation, and in particular, he pipetted all 3 phases. To measure all of the viscosity directly through pipette aspiration, which gives that the actin is much more viscous than the fluid, not very surprisingly. You can also measure the mesh size between two granules. The actin mesh we didn't measure directly because it's much smaller than what you can get with confocal mascoscopy. But the point is that as soon as you plug this number of magnitude, because the actin mesh is so much smaller than your granules, essentially you always have given these values, you always have actin fluid friction that dominates by at least an order of magnitude actin to your granules, and that's really because of the huge difference in size between the two structures. So that suggests that we should be here, right? That we should be in this region of the phase diagram, and therefore that the velocity of actin and fluid should be exactly the same, which you can validate. I mean, that's not so surprising, right? They went together, but actually also their average velocity indeed cannot be distinguished, which sort of validates that we're around here. And now the second question you can ask is, okay, but how do you generate these actin flows, right? Because otherwise, if you don't know how they work, we just displace the problem. And the advantage is that's pretty similar to what I mentioned before. Even though you consider a three-species medium, you still have a contractile instability, which is very similar, actually, to the one I discussed before, just for actin, right, where you have more actin makes more tension, more tensions recruits more actin. And actually, one of the interesting things that comes up out of the only thing that interesting things that come up from the interaction of granules and water is that you end up with a phase segregation which is always macroscopic, which is that you always end up with a single pattern, with a single accumulation of actin and fluid on one side. And so you can start, for instance, from very low noise, and you can input waves of contractility, and what you get is after each wave, you accumulate more and more actin and fluid, and that drives, and that's irrespective of system size. You're always going to get a single accumulation of fluid and actin in this kind of model. You can actually be a bit more systematic, so that's the parameters of the model. In principle, you could be more systematic because you might think that the granules are attached to actin actively through some active linkers, and therefore that it might be in some other settings the granules which are transported by the motors, and you get some interesting phase diagram, which is where the only two key parameters is the rescale contractility and the ratios of the friction coefficient. If you have much lower friction coefficient, if you have much higher friction coefficient for water, you phase segregate, and that's because you have a positive feedback loop. So if you actin brings fluid, but then the fluid brings monomers with it, so you polymerize even more actin. So that's a self-reinforcing feedback loop that gets phase segregation. But interestingly, if actin transports your granules, it's depleting the water that goes with it's depleting the water because you're enriching granules with actin, and because of this negative feedback, you end up having instead oscillations in this phase, which are just driven because of this frustration, because the actin keeps bringing next to it your granules that don't have any monomers to polymerize onto. So and we're looking systematically in this kind of phase diagram. And in a way, this is, as a small sort of theoretical side note, you could imagine this in any situation, right? I mean, this model is valid for any mix of water, active material, which could be cytoskeleton, but which could also be tissues, or any passive material. Here it's granules, but you could imagine the ACM for a tissue or organelles in a single cells. So in principle, you can get any kind of these things. But to go back to the key prediction, what we said is that the phase segregation should be really contractility dependent, right? It should be something that depends critically on your myosin concentration. And indeed, what Chaire did is to manipulate myosin contractility. And if you impair myosin contractility in this experiment, what you end up with is imperfect phase segregation. So you end up with lots of fluid, lots of fluid intermixed with the yolk granules. So that indeed it looks indeed like the phase segregation is contractility dependent, but also, and not surprisingly, this is because the actin velocity is reduced for lower contractilities, and as a good safety check, we can see that even in this condition, ooplasm flows exactly at the same speed and dynamics as actin, it's just that the two are reduced. Yeah, 3 minutes, okay. All right. So this is an explanation for this self-organized reinforcing, self-organized feedback loops on actin contractility. But now let's go back to control, right? Why do you always accumulate at the animal pole? There's no reason a priori. If you have something purely self-organized, there's nothing that would tell you that your fluid should always be there. And indeed, if you remember the simulations from before, you had an accumulation in the center, actually, right? There's no reason. If you remember the picture before, you had an accumulation in the center. Actually, if it's self-organized, you could accumulate in any place. So you need something to break symmetry. You need something to control your transition. So then we went back to what it could be, and actually a very simple thing, actually both at the cortical level, that's very obvious at the cortical level, but even also if you look at bulk actin intensity, is that prior to all of this, the embryo is heavily patterned animal-vegetal axis, right? There's way more actin both in the cortex and in the bulk in this pole than in this pole, and you can quantify this, and this is even before the actin waves. So you really have a pre-patterned gradient in the oocyte. So what we can do is then we can put this gradient in the model, and again, so the conductivity and all the viscosity and frictions were measured before through different experiments, and you can ask, okay, is this sufficient to robustly bias my self-organized flows? And it turns out that yes. So that's a chemograph where you start from noisy initial condition. So that's the AV axis, and that's time. Initially, you have uniform actin, uniform ooplasm, and what you get for every oscillation, you start transporting more and more fluid to the top, and you get also interesting coarse graining dynamics where initially in uniform things starts coarse graining into big pockets, and then each of these big pockets end up going up at every phase, right? And then we can, so this is in a way sort of parameter free, and we can compare it to the data. And this is a highly variable because this is highly variable as predicted. But actually, if you look at the data, right, you do see this phase of coarse graining when you end up with a few accumulations in there, and then these accumulations have these oscillatory dynamics with the correct slope, which is encouraging because the conductivity was taken from laser ablation experiment. So you seem to pretty accurately reproduce the full phase segregation data with the model. But to come back to before, the really key prediction is if it's self-organized, then if I remove this pre-pattern gradient, everything should be normal, but just mislocalized, right? This is back to this figure where my accumulation was in the center. So the key prediction if it's really a mix between self-organization and control is really this. So we thought, okay, let's try to play with actin dynamics to try to remove this pre-pattern. And one of the lucky things is that if you play with the intermediate dose of drugs which perturb actin dynamics, so either Cyto B or JASP, you don't change the bulk level of actin, but you do weaken the gradient. So you have with these simple perturbations different ways. I mean, the two actually change at different levels, but you have some levels of change of the gradient. And what was cool is that if you run the movie, what you end up with is, in both of these cases now, the fluid accumulation is either completely central here or heavily moved towards the center. So you can really show that indeed, if you perturb the gradient, you perturb the localization of the phase transition. But actually, in the end, you do get a pretty phase segregated embryo, right? So it's really a mixture between self-organization and control. And you can, again, do exactly the same simulation as before, so where you put a weak gradient, a weak pre-pattern gradient, and what you get is this phase of coarse graining dynamics and oscillations and oscillations. I'm almost finished. And you can compare this to the two dynamics in the two conditions. And again, this is pretty noisy, but in particular in JASP, I think you can see pretty quickly that you start with something pretty homogeneous, you end up with something coarse-grained, and you see this nice sort of slow coarse-graining through oscillation dynamics. This is all 1D, but we also looked at 2D dynamics, and I'm not going to go too much into the detail of how the 2D dynamics work in here because I'm out of time. And so just as a last thing, which I think is what I think might be a general principle in here because it emerged in many different ways, is that you have a mixture of self-organization that essentially powers your self-organization, which effectively powers your phase segregation, and then you use small guiding cues, which are enough because of the sensitivity of the phase transition to robustly localize, and in a way this bears a lot of resemblance, for instance, to the kind of mechanisms that are around cell motility, right? Self-organized flows with small gating gradients, or how Oocyte dynamics, for instance, in Drosophila, can bias self-organized actin flows. And this is also an example, I think, where you can you can model the system at mesoscopic scale, but that makes testable molecular prediction where you can go back and forth between theory and experiments. Yeah, so thank you very much for your attention. I just want to acknowledge Cholet, who did all the work in my lab, as well as Cheyenne and CP from the Eisenberg lab, as well as Pierre Réchaud, who's a long-term collaborator and with who we're working on this kind of model extension. And thank you very much for your attention.